So, good afternoon and a warm welcome to all of you here in the room and those joining us online. I'm Ursula Teuretsbacher. I'm an independent expert for R&D, antibacterial drug R&D with a focus on public and global health aspects. My co-chair is Keith Klugman. He's very well known. He's the pneumococcal guru and now chairing and leading major programs at the Gates Foundation. So this opening symposium focuses on the critical issue of antibacterial drug R&D with, of course, specific emphasis on the unique challenges of treating children, as well as the global and also regional variation in the scale of the problem of resistance, which much of it is caused by political and socio-economic factors. We will explore several of these factors and also suggest some potential solutions to mitigate the growing burden of antibacterial drug resistance. This session will consist of 4 brief presentations followed by a panel discussion. So please hold your questions until the last presentation. You can submit your questions using the Q&A function in the conference app under Interactive Features, located next to the session title. Our online audience may use the Q&A panel on the virtual platform to submit questions throughout the session. Before we begin the scientific program, I kindly ask you to silence your cell phones. With this, I want to invite our first speaker. It's Ramanan Laxminarayan. He's founder and president of the One Health Trust in Washington, D.C. and Bangalore, a senior research scholar at Princeton University, and director of the WHO Collaborating Center on Antimicrobial Resistance in New Delhi. He will talk about the importance of appropriate access to effective antibiotics. Please, Ramanan.
Thank you, Ursula. It's a real pleasure to be here, and thank you both for inviting me to do this. Most of what I'm going to talk about is really from this Lancet series on affordable access to effective antibiotics, and actually it needs to be sustainable as well. There are a couple of copies out there in front, or there are also a bunch of summaries out there of this, so if you're interested and you can pick it up. Of course, the Lancet series is also available for free online at the Lancet website. It came out, I don't know, roughly a year ago. I don't have any conflicts to disclose. Like I said, this all comes out of that series, what I'm going to talk about here. I think, obviously you're all at ESPID, you really don't need an introduction to antibiotic resistance, but I think to frame the issue, we need to remember that the problem we're tackling is not AMR first and foremost. It is the 7.7 million deaths that are associated with bacterial infections, of which about 5 million, less than 5 million, are associated with drug-resistant pathogens. And then a subset of that, 1.27 million, is the one that is actually attributable. Now, obviously, these numbers, which come from the University of Washington, have they based largely on data from developed countries. There's not a lot of developing country data behind it, but it also tells us that we need to be paying attention to getting better data so that these estimates can actually get better. Now, at least in high-income countries, resistance to third-line antibiotics could be 2 times higher in 2035 than 2005. That's from an OECD report. And the impact is heaviest in low and middle income countries. If you look at deaths per 100,000 population, most in sub-Saharan Africa, followed by South Asia, not too far behind. And then you have, interestingly, Central Europe, Eastern Europe, and Central Asia, which is where we are right now. So the burden is in every part of the world, but obviously concentrated in places where there's a huge background of infectious diseases as well. And there are many reasons why this is important. One being that if we care about the Sustainable Development Goals, you all saw the slide on that as a precursor to this up there. And the SDG goals on child survival would be much easier to meet, if we had access to effective antibiotics. So the blue line, the dark blue line is the actual, and then if we didn't have AMR, it actually cuts the gap between where we are and where we need to be. And it's true for newborns and also for children under the age of 5. It also threatens progress on healthy aging, which, as it happens, is one of the Sustainable Development Goals. And these pathogens are in every part of the world, but as you can see, healthy aging is particularly a problem in South Asia because of bacterial pathogens that don't respond to antibiotics. Now, this slide probably needs to be updated for the events in the United States over the last 4 months, but this was what was published in The Lancet. I think we have substantially less money for TB and HIV compared to what we did when this was published. But enough to say that $2 billion spent on AMR every year, which includes literally all spending on R&D, regardless of whether it's happening in the public sector or the private sector, is totally is not commensurate with the burden of drug-resistant infections. And this is where you all as a community are part of this advocacy, which is to make sure that people understand that we don't have the R&D to tackle AMR. And this is not just about new drugs, it's also about vaccines and diagnostics. Now, the number of drugs approved by the FDA seemed like it was dropping for a little while, and then it's going back up again. This is a picture that Ursula, who's one of the co-chairs, really hates because it doesn't in any way correct for the quality of antibiotics. Many of these drugs are just Me Too drugs, so just saying we had new antibiotics, or we had 8 new antibiotics, since 2020 doesn't really make any sense at all, because it doesn't tell us if it's matching the burden of infections. But that said, what we do know is that when companies invest in antibiotics, it's a definite path to failure and bankruptcy. Now, the reason for that is that these drugs typically are first sold in high-income countries at very high prices. I'll show you a slide on this shortly. And they are sold in countries where there's not a high burden of drug-resistant pathogens. So you have, on the one hand, LMICs where all the patients are, and then you have the drugs in the high-income countries where there's just not the same number of patients. The folks in the LMICs can't afford these drugs if they cost what they do, and the folks in the high-income countries also have options. So it's a question of a product mismatch with the geography in which they're being sold, and you don't have to be a drug developer or a commercial person from a pharma company to know that if these are your sales of an antibiotic, which you've spent probably close to $1 billion on, you're not going to succeed. So many of these are just cut off, and ironically, in most pharma development, once a drug is approved by FDA, their share price goes up. And in the case of antibiotics, the share price drops immediately as soon as it's FDA approved, because that's when the investors wake up and realize there's really no money to be made. Now, even when the drugs are actually produced and are registered, they don't reach the populations that need them. This was a study we did a few years ago, which looked at the total number of drugs which were developed but also made it outside of the 10 countries, and you can and you can see that there are big gaps, light blues in most parts of the world. The only places where the drugs make it are in North America, probably Brazil, and China to some extent, India, because some of these drugs make it there as well. But even there, most drugs, over 60%, don't make it out of more than 10 countries. So when we're talking about R&D, the fact that we have research and development in in a company doesn't mean that the drug actually reaches the countries. So that is the first step to access. If you don't have it reaching the country, then you don't really have access at all. And my colleague Peter Beyer is going to talk about GARP and what it does to make sure that we're solving that problem using public funds. Also, the drugs that are developed, this is slightly dated, but gives you a sense of what's happened in the past, and it's not very different from before. Even between 1980 and 2009, which we think of as a golden, not a golden period, but at least not a bad period compared to what followed for antibiotics, about 43% of drugs were withdrawn either because of toxicity or lack of market, compared to a 13% withdrawal rate for other therapeutic categories. So this is a very challenging place to develop drugs. Now, we did have an earlier version of this Lancet series back in 2016. It was launched in 2015 in time for the 2016 UN General Assembly high-level meeting on AMR. And one of the things that it helped capture was this idea that we have three legs on which our response on antibiotic resistance should be. The first is access, because there are millions of people without access to antibiotics. We also need conservation of existing drugs, and then we also need innovation in creating new antibiotics. So we need all three of these. Now, if we have access without stewardship and innovation, that just speeds up resistance. So in other words, access is a good thing, but it does come with its challenges. You need to come up with new antibiotics. If you only conserve, in fact, the best way to prevent resistance is to not use any antibiotics at all ever. Now, that would obviously not be a very clever thing to do, but when we do stewardship without thought, we constrain access. So stewardship which is not thoughtful about access is actually quite harmful, but it also undermines innovation, because which company wants to make a drug that is just not being used at all? So this needs to be thoughtful with respect to those two incentives. And of course, innovation alone without access is unjust, and without conservation, it's wasteful. We do need to, somehow square these three concerns. And I think, I used to present this very slide, 15 years ago, and I almost never thought that this was really going to be the way in which things worked out. Of course, one bit I added, which is, so penicillin is a drug which is still, pennies. Linezolid about, $155. Daptomycin, 2006 it was introduced, $181. Cefiderocol, 2019, so it sells for about $4,800 in the US. So the point that was being made here was that the price of drugs has been steadily going up over time, but even so, it is nowhere close to what the newest cancer drugs that prevent someone from dying for just about 3 months. So this particular drug extends life by 3 months. So if there are people willing to pay $10,000 a month just for extension of life, imagine the value that antibiotics bring to the table. But it also warns us that if we're not careful, we're headed for those kinds of prices for antibiotics as well, which would put them well out of the reach of most people in the world. So there's a cautionary tale here, which is that the market is telling us something about this, which is there's a value to life which is quite high, but at the same time, we shouldn't take the continued affordability of antibiotics for granted. In fact, this was a picture that we made for this Lancet series. And what you can see is these are the costs per treatment course in US dollars. And look at all the costs prior to 2005. They're probably $100 at the most. After that, it just takes off. So after 2005, the drugs that have been introduced cost anywhere between $5,000 and $15,000 per treatment course. Now, I know many folks in this audience are quite young, so maybe this seems like a natural thing to be, but you also know that there are antibiotics that are, not more than $5 or $10, and that's really what most antibiotics are supposed to be in a sense. But that is not the future we're headed towards at all if you look at innovation. So we really have a challenge here in terms of access. I'm not so worried about R&D, because I think there will be research and development in drugs which are very expensive, that are limited to just a few patient population. It's for the rest of the population that cannot afford these drugs that is really the problem. And so greater access to antibiotics is ironically essential for curbing drug resistance. And you say, why? Because if we can serve people in low and middle income countries, we can probably ensure that there's a market for antibiotics that ensures that all drugs are actually inexpensive. And companies perceive the potential revenues and market size in LMICs as insufficient to justify the cost of market entry. But the patients most in need are in these countries, and in fact, that is the solution to actually solving this problem of AMR. Now, 2024, we had a UN high-level meeting in New York. There was a declaration, and in the lead up to the declaration, all the political sort of discussions that happened, this is a large part of where the challenges were. High-income countries were very focused on antibiotic resistance. Low and middle-income countries were largely focused on access to effective antibiotics. And this is where there was a huge, debate about, where we were headed. Because since 2016, LMIC saw most of the money had gone just to new drugs, nothing to improve access in any way at all. And this is a point I can't stress enough, which is that access to effective antibiotics, or the lack of access to effective antibiotics, still kills far more people in the world than does resistance. So you go back to the very first slide that I showed, 7.7 million deaths because of bacterial infections, 1.27 because of drug resistance. So there's a lot of people who are treatable with antibiotics that exist today. We don't pay attention to those. We're paying attention to those ones that are not treatable, but why not also pay attention to those that can be treated with the antibiotics that we have? And access to effective antibiotics should really be our lens into how we look at this issue, and not just use resistance as really being, the way to be, to drum up, interest in the issue. Because 7.7 million, just for comparison, at the height of the HIV epidemic in, say, 2001, 2002, when the most number of people are dying of HIV/AIDS, the total number of deaths from HIV globally, does anyone know what that was? 2001, that's like peak HIV deaths. It's about 2 million deaths. 7.7 million is almost 4 times 2 million. But we don't see that outrage about bacterial infections. There's no activists, there's no pins for, no pink ribbons for bacterial infections. I'm not downplaying HIV. I think HIV and TB are terribly important, but I think bacterial infections are also terribly important. Now, what we did in this was also then not to focus just on burden, but also look at avoidable burden, which is very important, because one of the best things we can do to prevent resistance is by not having an infection in the first place. So this was to look at water and sanitation, all wash, hand washing across low-income countries, lower middle countries, and upper middle-income countries, and to estimate the total number of deaths that you could avert by investing in those strategies. So this was a fairly detailed piece of work that was done. And if you look at the AMR mortality averted by scaling up IPC, infection prevention control, wash, and vaccine interventions, you can see that you get the biggest bang for the buck actually in lower middle income countries, because IPC is important in places where people are actually ending up in healthcare facilities. In low-income countries, not enough people are in healthcare facilities. In upper middle-income countries, they've got a handle on how to do this. So we have a huge opportunity in sort of that middle range when people are going to hospitals, they are getting infections, hand hygiene is important, but vaccination coverage is also really important. You'll hear from Dr. Padmini Srikantiah on vaccines and AMR, which I think is probably the most useful and important thing to do when it comes to reducing the burden of drug resistance. In fact, I just came from a meeting on animal vaccines, and it's the same story there. Animal vaccines are the best way in which we can reduce antibiotic use in animals, and I think it's true for humans as well. And this picture shows rates of antibiotic use averted by interventions. Since 2018, we've now have a significant number of studies that are able to document the avertible antibiotic use and the avertible number of deaths and DALYs from antibiotic resistance quite well across a range of pathogens. And this is something we didn't really have about 6 years ago. I think these are very useful advocacy tools in order to promote the use of vaccines and prevention in order to tackle the problem. Now, so to summarize in the last couple of slides, you have about 337,000 deaths that can be averted by aligning IPC standards, access to wash services, another 250,000 deaths, and high priority pediatric vaccines that already exist, about 180,000 deaths. So in total, about 750,000 AMR associated deaths are avertible each year in LMICs just through interventions that we have right now, we know how to do, And in many cases, we even have the money to do. There is no reason why these 750,000 people should be dying every year. It's simply because we have not really paid attention to prevention as really being the main thing we can do about AMR right now. Now, building on this, we called for, in the Lancet series, a 10, 20, 30 targets. Now, the 10 and 20 made it into the final declaration. The 30 got changed to something even better than 30. So the 10% reduction in mortality from AMR is actually now part of the UN declaration. By 2030, relative to 2019, we would achieve these targets. So it's something that all countries, including Romania, have signed on to. 20% reduction, actually it's a 20% increase in access antibiotics, which is sort of a proxy for this measure. And the declaration calls for a significant reduction in inappropriate animal antibiotic use. By significant, I take away that it's at least 30%. They didn't want to put a number there, but you can't say 5% is significant. So I would say 30% is probably significant. So in some sense, the things that we called for in the Lancet series all made it into the final declaration. And they're also well within the reach using existing interventions. So we then work backwards to say, to get those targets, 14.2% of AMR-associated deaths in LMICs are avoidable if 20% more healthcare workers practice hand hygiene. Do we think we can do that? 20% more health workers? I think that's possible, right? Can we have universal pediatric vaccination coverage? And we can do the rest from this as well. So the last slide is really to say that we really want to have an independent scientific body to expand the evidence base and increase funding, especially for infection prevention programs. And all of these should encompass both human and animal health. Thank you very much for your time. And I'll stop here.
Thank you, Ramanan.
So I just want to make a request to Kenneth. There's an unacceptable amount of noise in this room, and it's people who are in the corridor walking behind the room. So if Kenneth can go into that corridor and move people out, or have signs and say silence for people walking around the room, because I don't know if you've noticed, but it's very, very loud in here.
So, thank you for this very important note. The second speaker is Peter Beyer. He's a trained lawyer and public health expert who joined GARD-P from the WHO, where he led initiatives to develop and improve access to new antibiotic treatments. He will talk about the Global Antibiotic Research and Development Partnership, that's GARD-P, a new approach for R&D and access. Please, Peter.
Thank you so much, Ursula, for having me. It's a great honor to be here. And I have no conflict of interest to declare. And now I'm going to give you an introduction into GARD-P, but also the work we do specifically on neonatal sepsis. So we are following a non-profit approach to antibiotic R&D and access. We are a Swiss-based foundation, so we are a not-for-profit organization. We have about 100 staff globally, and we have about an annual budget of 35 million. So why do we engage in children antibiotics? Because we do not believe that the market is going to fix this problem. I mean, Ramanan spoke very eloquently on the lack of actually antibiotics for adults, but it looks actually much worse actually if you look at the pediatric formulations. What we are doing is we are focusing specifically on vulnerable populations. That is why we look into children and newborns, and we want to accelerate pediatric formulations so that children and newborns have quicker access to more effective and safer treatments. And of course, very important, Ramanan spoke about it, our mission is also to improve the access and to ensure access to the treatments that we are developing. This is a quick overview on the current portfolio. We are expanding. We are going to open a couple of new, very interesting R&D projects. But here you can see that for carbapenem-resistant infections in adults, we have a license to cefidorocol. It's a reserve antibiotic. We have the license for 135 countries, and we are currently transferring the manufacturing technology from Shionogi in Japan to our manufacturer in India, Orchid. And we're also going to work potentially on the pediatric, on the neonatal sepsis program. I'll speak about it later. Another drug in our portfolio is cefepime taniborbactam for carbapenem-resistant infections. We developed this with Venetorix. This is currently on clinical hold. Unfortunately, if that is lifted or if that proves to go to registration, we would actually ensure the pediatric drug development. And then this compound BVC 0977 is owned by an Indian company, Bugworks, with whom we have a collaboration and license agreement. We are currently restarting the phase 1, so you can see it's a very early drug development project. And then in neonatal sepsis, I'm going to speak in detail about this empiric treatment regimen that we are trying to develop for neonatal sepsis. And in sexual transmitted infections is our third area. We hope that our partner is going to submit to the FDA the dossier for the approval of zoliflodacin for gonorrhea infection. We have done, as a foundation, the biggest gonorrhea trial with nearly 1,000 patients in 5 countries and showed non-inferiority to the current standard of care. So we hope to make this available soon, starting with Thailand and South Africa. So this is what we did when we started the work on neonatal sepsis. We started actually with a global observational study to look into the data, and we enrolled more than 3,000 newborns in 19 hospitals across 11 countries. We started in 2018. You see, in 2020 we completed enrollment, and the data was published in 2023 in PLOS Medicine, together with our key partners. We found that there was an average mortality of about 20%, but if you look at the different sites, you had sites where the mortality was below 2%, and we had sites where the mortality was like 27%. We found a huge amount of infections due to carbapenem-resistant pathogens, in particular Klebsiella. And what was, I think, most surprising is that we saw that doctors were using more than 200 different combinations of existing antibiotics to treat neonatal sepsis. And most of these combinations, there's actually no data on what is the correct dosage for neonatal sepsis, and actually whether the combinations are synergistic or antagonistic. And that's based on this data, we identified 3 potential new treatment regimens: fosfomycin with amikacin, fosfomycin with flomoxef, and flomoxef with amikacin, which we believe, based on the data and hollow fiber studies, to be actually active against the carbapenem-resistant pathogens and be new treatment options, in particular for those sites where actually resistance is very high. And that is why we started the NeoSep clinical trial, where we have a part 1, which is a pharmacokinetic phase of new combinations to prove the safety and the dosage, and then the part 2, where we are going to look at actually the new treatments, including the existing treatments. So this is the part 1 we just finished. We did 3 sites in Kenya and South Africa. In Kenya, it's the Kilifi County Hospital, and in South Africa, we did it in the Chris Hani Hospital in Soweto, and Tygerberg in Cape Town. And I'm really grateful for these sites that they actually collaborate with us. It is a fantastic collaboration with these hospitals, and they enrolled overall 65 babies to confirm the pharmacokinetics and also that there's sufficient exposure for bacterial killing, which allowed us to move forward with the part 2, where we are testing these 3 new combinations against 5 additional regimens that are also already used by clinicians. And so this clinical trial design is quite innovative. We call it practical, and because it is a randomized trial where we actually each patient is randomly assigned to a clinically appropriate regimen, versus you would actually test one regimen against standard of care. As I said, in the observational study, we found more than 200 different regimens being used. So basically, there is no standard of care. And so you can't actually, and also if you would test a new treatment regimen against standard of care, you would only generate data for one actually potential new treatment regimen. With this practical clinical trial regime, trial design, we want to generate actually data where we can rank these 3 new treatments and the 5 existing treatments, and so we can rank it to see which ones actually perform better than others. Because we do not believe that for these, for all regions in the world, you can find the one actually optimal treatment, but you can identify which are the best treatments for these babies. And that is what will be the outcome of this trial. And of course, what we are also going to do once we are finishing the trial, you can see now we opened actually the site in Tygerberg, where we already did the part 1. And here you can see how we want to expand in this year in Ghana, in Kenya, in Uganda, so more sites in actually sub-Saharan Africa. And then we are going to move to Asia, Bangladesh, India, Malaysia, Pakistan, Vietnam. And you can see here that this is going to be a long trial. So if you can, I mean, if you saw that in 2018 we started the observational study on neonatal sepsis, these results are going to be published in 2030. So it takes you 10 years actually to do this kind of work. And then, of course, as I said, we are also going to ensure access to these new treatments. We are already working with the manufacturers and potential new manufacturers for fosfomycin and flomoxef. And we also are going to work with WHO that they hopefully are going to update the guidelines once the data is out. This is a slide on our general work on children antibiotics. So what we want to do, I mentioned it for cefepime taniborbactam, we want to contribute or carry out the pediatric development for new antibiotics and to accelerate the development of appropriate formulations. And we are working very closely with the Global Accelerator for Pediatric Formulations, GAPF. I'm sure Valeria is going to talk about it because it's a WHO-led program. Briefly on cefiderocol, pediatric development update. So what we are doing, as I said, we have the license to this reserve antibiotic for 135 countries. It's registered in a number of countries already. Shionogi has completed the pediatric regulatory program, and they have established actually the data for safe neonatal doses. And we are now considering to do further studies to support the appropriate use to treat children and neonates in settings with carbapenem-resistant infections. And so this could include additional PQ studies, as we did in the part 1 of NeoSep. But we're also considering enrolling cefiderocol as another treatment option in the neonatal sepsis study, which could be either in monotherapy or in combination. As I said, this study, NeoSep, if you include the observational study, it's taking us about 10 years probably to do this from the start and to the end. And I also wanted to highlight that the observational study, the costs were about 3.8 million, and the NeoSep 1 with the part 1, part 2, we already spent about 7 million, and we expect that it's going to cost another 38 million. So basically the project is 50 million over 10 years, and here you can see this is the funding we have secured so far. Thank you so much, Kees and Padmini, for your support. Fortunately, neonatal sepsis is one priority of the Gates Foundation. I'm responsible for fundraising in GARDP, and I can tell you it is counterintuitive, because you would think for neonatal sepsis, for pediatric formulations, it would be much easier to fundraise than for other programs. But the reality shows that this program is actually not easy to secure the funding that you need. And one reason is actually what we believe is the strength of the program. We believe the strength of the program is that we use old antibiotics, which are generic and cheap to manufacture, and for which we already have actually good safety data, and that we know we can actually use in neonates, and where in a certain, a relatively short period in time, we can actually develop new regimens and come out with the data. But it means, of course, also that this is not innovative, as you would call it. And that sometimes makes it more difficult to actually find funding, because funders often like the innovative new breakthrough drugs, like our drug for zoliflodacin is a new class in antibiotics, and it makes it's much sexier to say we actually register the first agent of a new class of antibiotics. But in reality, for the patient, it doesn't actually make a difference. And maybe they are better off in taking the old antibiotics, for which we have much better data over the years. And we believe also that with this public money that we are investing, it is much cheaper to invest this money in making better use of the current portfolio of antibiotics we are having, that we are underusing and we don't use appropriately, because we are missing actually the clinical evidence on how to use it in the patients. And that is something that we want to do, and why we have been set up. For example, we want to do a similar study in ESBL infections in adults in Japan, Korea, hopefully Saudi Arabia, that we are currently preparing together with Advance ID, where we want to have a comparable clinical trial design. And we also want to test the same agents and some other agents to see whether we can actually develop a carbapenem-sparing regimen. And with this, I think I can give it back to Ursula and Keith.
Thank you very much. Thank you. Thank you very much, Peter.
So it's my pleasure now to invite Padmini Srikantiah, who I'm very privileged that she works with me. She's a deputy director at the Gates Foundation. Padmini is ex-CDC. She's an infectious disease physician. And she's going to talk to us about vaccines against AMR. But before she starts, I want to thank Kenneth for lowering the voice level in the room, and thanks Ron and Oana. I saw you also helped with that. So I think it's better. It's not perfect, but over to you, Padmini.
Thank you, Keith. And it's my great pleasure to be with you all this afternoon to speak in this session, thanks to the organizers. And I have no disclosures or no conflicts. So I'm going to start with the figure that you saw in the first talk with Ramanan, which is really about the global burden of antimicrobial resistance. And the updated numbers from the 2024 Lancet paper that the Institute of Health Metrics and Evaluation published indicated that there are 1.1 million AMR attributable deaths, or estimated, and this is out of a total of 4.7 million AMR associated deaths. But as Ramanan mentioned, I would like to also underscore that this is all really driven by a burden of bacterial infection. So this larger number, which is 7.75 million, and really what I'm going to focus on is that the prevention of these bacterial infections and the related antibiotic use is key to addressing AMR-associated deaths, as well as helping to reduce antimicrobial use. Because this is a pediatric infectious disease conference, I've focused this map on AMR-associated deaths in children under 5. And if you look at the left figure first, this is the total deaths per 100,000 population of all pathogens in children under 5 who have bloodstream infections. And you can see this heat map scale on the bottom, where the greater burden is reflected in the red and yellow colors. And you can see where the greatest burden of bacterial deaths from bloodstream infections in 2021 lies in these low and middle income countries, especially in Western Sub-Saharan Africa. Not surprisingly, the total deaths per 100,000 population that is associated with AMR in 2021 in the under 5 population is similarly in the same geographic area, which to me reflects that there is really a great overlap between the AMR-associated deaths and deaths due to bacterial infections and the bacterial burden of disease. This area in Western Sub-Saharan Africa does not, if you were to measure antimicrobial use, is not higher than others in terms of antimicrobial use, and rather this burden of AMR-associated deaths is a reflection of bacterial infectious disease. So getting at the heart of these bacterial pathogens and trying to prevent them is a key next step or a key step to addressing AMR. So let's take a closer look, again in the under 5 population, at the pathogens causing AMR-associated deaths. In 2021, there were an estimated 840,000 AMR-associated deaths in children under 5 years of age. And I will point out that children under 5 years of age account for about 7 or 8% of the global population, while this AMR-associated deaths number is about 18% of global AMR-associated deaths. So this under 5 population is disproportionately impacted by AMR. I hope you're able to see the pathogens on this. I'll have to wear my glasses to read it. But in terms of the top 10 pathogens, which account for the great majority of these 840,000 deaths, led by Streptococcus pneumoniae, Klebsiella pneumoniae, E. coli, Staph aureus, Pseudomonas, Acinetobacter baumannii, Group B strep, Serratia, Salmonella typhi, and Shigella. Vaccines are currently only available for 2 out of these 10 pathogens. And so we have some work to do, and also we need to reflect on what have we learned from vaccines that are addressing these 2 pathogens, and what can we take away from that for vaccines that address potentially other pathogens. So some years ago, we supported some work, and I'll talk a little bit about some of the modeling work that we supported Ramanan's group to do, where he developed an AMR and vaccines modeling consortium. But we also supported WHO, who then developed a WHO framework for vaccines to address AMR. And as you can imagine, vaccines could contribute to the reduction of AMR in a number of different ways. They can prevent infections due to both drug-susceptible and drug-resistant pathogens. That can directly protect the individual from getting sick, as well as safeguard the communities from getting sick, prevent complications, and the decreased infections means potentially decreased antibiotic use. And there's the potential to also have suppression of resistance evolution and decreased transmission of resistant pathogens. Now, importantly, the impact of vaccines against antimicrobial resistance is really vaccines against infectious diseases, so reducing infectious disease burden. And this impact can not only be yielded from vaccines that target bacteria, but also against vaccines that target viruses, for which we're not necessarily, obviously not measuring antibacterial resistance, but these viruses can lead to an excess of antibiotic use, and vaccines that target viruses can also play a role. So let's look at some of these key pathogens. The first I'll look at is pneumococcus and the impact of pneumococcal conjugate vaccine on penicillin non-susceptible strains. So the figure on the left is from South Africa, where they introduced the pneumococcal vaccine around about 2008. And you can see in yellow the significant decline in penicillin-resistant invasive pneumococcal disease, an 82% reduction. In addition, in green was the multi-drug resistant strains of pneumococcus, and in blue is 85% reduction in ceftriaxone non-susceptible strains. So obviously we know that the pneumococcal vaccine works to prevent pneumococcal disease, and it works just as well to reduce those strains that are penicillin non-susceptible as well as ceftriaxone non-susceptible. This introduction of PCV also was associated with a reduction in antibiotic use. Now, we have seen some of these gains, and I've given you an example in South Africa. Some of the modeling work that Ramanan's consortium did indicated that if we actually improve the coverage of pneumococcal vaccine to 90% of those who need it, as well as in the elderly, we could avert an additional 27,000 AMR-associated deaths and 1.3 million DALYs, and prevent quite a substantial cost in terms of hospital costs. And this, I'm sorry, actually comes from a WHO vaccines and modeling, vaccine and prioritization framework report, which I'll describe a little bit more later. So let's turn now to another bacterial pathogen for which we're concerned about multi-drug resistance. And this example comes from Pakistan for Salmonella typhi, where some of you may be aware that there has been a problem of multi-drug resistant and indeed extremely drug-resistant Salmonella typhi in Pakistan over the last several years. It was due to this outbreaks of extremely drug-resistant Salmonella typhi that the Sindh province in Pakistan introduced the typhoid conjugate vaccine, and they evaluated this, the effect of this typhoid conjugate vaccine on both on culture-confirmed cases of Salmonella typhi. And what you can see is, of course, there was significant efficacy or high efficacy against all culture-confirmed Salmonella typhi cases, and indeed similar efficacy, over 95%, against extremely drug-resistant cases of Salmonella typhi. So we know that these vaccines, when they work, they can work just as well against these extremely drug-resistant strains. So what happened in Pakistan? So this slide is thanks to Dr. Farah Qamar from the Aga Khan University in Pakistan. And she presented these data a few years ago in another meeting, where if you look at the blue line, or blue vertical dashed line, which is when they held their TCV campaign between 18th November and 7th of December, subsequently you see the significant decline in typhoid cases. However, I think what is interesting, and we'll have to see over time how this plays out. In the red line, you see that the proportion of typhi cases that are XDR has not significantly changed. It still remains over 50%, while there is an overall significant decline in all typhi cases, including that of extremely drug-resistant and multi-drug resistant strains. So we do see this impact on typhi. We would like to see longer data in terms of what this means for the proportion of extremely drug-resistant or multi-drug resistant strains. I'm going to turn now actually to two vaccines against viruses. Another vaccine against a diarrheal disease pathogen, rotavirus. So this actually is a paper that came out from the Vaccines and AMR Modeling Consortium that Ramanan coordinated over the last few years. And they looked at antibiotic-treated diarrheal illnesses in children 0 to 23 months of age. In all low and middle income countries, the incidence of antibiotic-treated diarrheal illnesses due to our rotavirus was estimated to be about 20 per 100 children. And the total number of estimated antibiotic-treated illnesses due to rotavirus in the absence of vaccination was close to 50 million episodes of antibiotic-treated illnesses. In the 2018 coverage, this paper came out in 2020, in the current coverage of rotavirus vaccine in 2020 indicated that there was a 13 million antibiotic-treated illnesses that were prevented under that current rotavirus vaccine coverage. If we were, similar to the point we made for pneumococcal vaccine, if we were to actually improve that rotavirus vaccine coverage to universal coverage, we could avert an additional 18.3 million antibiotic-treated diarrheal illnesses, especially in low and middle income countries. I'll turn now to RSV, a newer vaccine, and in this case actually a monoclonal antibody that I'm using as an example, but a prophylactic monoclonal antibody. And we see from the data that were published from a pooled analysis of their phase 2b and phase 3 trial data of nirsevimab, which is the long-acting monoclonal antibody that many of you know about and some countries here have started to roll out, that not only was it highly efficacious in reducing RSV severe disease and indeed all-cause lower respiratory tract infection, but we actually see a 23% reduction in any antibiotic prescription in the first 6 months of life, where RSV disease and these viral illnesses are very common. They're a common reason for infants to present either to a clinic or indeed to a hospital and result in antibiotic use that may or may not actually be warranted. Prevention of RSV, indeed, in and of itself, may also help to prevent the secondary bacterial infections that may occur in severe RSV cases, and that also is likely contributing to this reduction in antibiotic prescriptions and use. So I've talked to you now about 4 vaccines, 2 for bacteria and 2 for viruses. All these vaccines are now available for use in many countries. We hope that RSV now comes to low and middle income countries in greater numbers in the next couple of years. But what about the significant AMR priority pathogens as we look forward? If you remember that list that I read off in terms of the top 10 pathogens that are associated with AMR deaths, number 2 on that list is Klebsiella pneumoniae. And you heard about Klebsiella pneumoniae. Don't know what happened. Can I get some help here? The screen has gone blank here as well.
I guess you'll just have to tell them the cool story.
So Klebsiella pneumoniae, there you go. So Klebsiella pneumoniae, as you all know, is a gram-negative pathogen. We see it as a priority WHO AMR pathogen with highly drug-resistant characteristics. In IHME's estimation in 2019, among neonates specifically, Klebsiella pneumoniae was associated with over 120,000 deaths. This is both due to susceptible as well as resistant infections. But the great majority of these infections, and these deaths indeed, are AMR related. Almost 78,000 or almost 80,000 AMR associated deaths, and of these 33,000, close to 34,000 that are AMR attributable deaths. So of course, we need the right treatment to treat these neonates who develop sepsis, and that's the work that Peter described from GARD-P. But we also need to think about how we can prevent these infections. And one way that we at the Gates Foundation are working on, and I know some others here in this audience are also interested in, is the potential of using a maternal vaccine strategy, similar to the RSV maternal vaccine that's also available, to use that maternal vaccine strategy for a pneumococcal conjugate vaccine. So pneumococcus, and I won't go into great detail here, Klebsiella, like pneumococcus rather, has capsular and subcapsular antigens that can serve as targets for a polysaccharide conjugate vaccine. And modeling work both in WHO has done and Ramanan's consortium has done indicate that a vaccine that has high valency and covers about 70% of the strains with 70% efficacy could avert more, potentially up to 80,000 neonatal deaths each year. Now, right now, we're years away from that potential impact. We're at the preclinical stages, and we're working on establishing preclinical proof of principle for a 10+ valent conjugate vaccine. Lastly, I'll just mention that WHO, in the work around vaccines and AMR framework, published a landmark report last year in November, which was around estimating the impact of vaccines to reduce AMR. And if we just talk about the existing vaccines, we could avert 106,000 AMR-associated deaths, and you can read these numbers in terms of quite a significant number of DALYs, as well as healthcare costs, in addition to helping to reduce antibiotic use. Vaccines in late-stage clinical development could avert an additional 135,000 AMR-associated deaths, and vaccines in early clinical development, such as what we're talking about with Klebsiella, where there is one candidate that has gone to clinical phase, could potentially avert an additional 400,000 AMR-associated deaths. So there's challenging work to do to improve that potential impact of AMR-associated deaths. So I'll finally close by reminding you that AMR-associated mortality, particularly in low and middle income countries, is truly driven by a high infectious disease burden. Vaccines can reduce this infectious disease burden and have potential to impact AMR through preventing bacterial infections, including those due to resistant strains, as well as preventing high-incidence viral pathogens like RSV and rotavirus, which can result in a reduction in antibiotic use. For pathogens like the ones that I talked about for which there is an existing efficacious vaccine, expanding coverage is critical to further reduce both disease, deaths overall, as well as AMR-associated deaths, and development of vaccines targeting AMR priority pathogens like Klebsiella pneumoniae is a challenging priority that could impact AMR-associated deaths and related economic burden. Thank you so much for your attention.
Thanks, Padmini, and we've given you a feast of information so far, but one of the things that has not been addressed is the question of antibiotic regulation in kids and how that can be improved. And so I'm delighted that we have Valeria Gigante with us. Valeria is a clinical pharmacologist who leads this program at WHO of regulation for antibiotic use, and she's going to talk to us about accelerating optimal approval of priority antibiotics in children. Now, as you're all aware, WHO is in the midst of major reorganization, so I'm very grateful to Valeria that she's able to talk to us from Geneva. Valeria, it's over to you.
Thank you so much. Thank you, Espid, for the invitation. It's really an honor to be here and to talk about some of the WHO research and development activities that I lead. Today, my presentation will also include some a
Valeria,
preview
can you talk a little closer to your mic if possible? Because you're a little faint.
So I just have a computer, I don't have a mic, but I will try to do my best. I hope that is working. Can you hear me better now? So my presentation today will also present some key results from the unpublished 2025 WHO antibacterial pipeline report with some concerning pediatric antibiotic development. And we will see together why there is the need to accelerate the research and development for antibiotics use in children. WHO support research, development, and innovation, identifying unmet public health need, but also identifying trends, opportunities, and prioritizing key research areas. With my unit, we establish global priorities, such as the bacterial and fungal priority pathogen list. And against this priority, we regularly analyze the pipeline of antibacterials, bacterial vaccine, antifungals, and from this year also on diagnostics for bacterial and fungal infection. I will present more in detail the WHO work to support the pediatric development, the DMR division lead with the GAPF consortium that is hosted, is a partnership hosted here in WHO. And from our side, I also lead the development of targets for R&D, including target product profiles. Last January, I had the pleasure to host the WHO Advisory Group on Research and Development, and the recommendation that we received to strengthen the routine pipeline of antibiotics talk about prioritizing innovation, improve access, providing funding to a small and medium enterprise, but also to increase pediatric development that lags behind the antibiotics for adult use of approximately a decade. Antibiotics are the most prescribed medication in children, but many of them lack authorized indication or optimal formulation to administration across the different age groups. And this causes medication error and also challenges. And in case of antibiotics, we have toxicity, but also the increase of resistance. And all this found some roots in the thin pipeline that we are continuing to see. And here for the SPID audience, I have a review of the key findings about the pediatric antibiotics currently in development. So we have 12 antibiotics in late-stage development from phase 2 onwards. And the majority of them do not present a pediatric investigation plan yet. And this data is consistent with our latest analysis published in 2024. Of the antibiotics authorized, so already available on the market, of the 17 of them, only 9 have an ongoing pediatric investigation. 3 of them are fluoroquinolone, so not indicated in children, and 1 of them, plazomicin, has an old pediatric investigation plan, so probably will not result in any pediatric studies. To advance the pediatric antibiotic agenda, WHO is working closely with the GAP accelerator, as I mentioned, across several pillars that includes research prioritization, R&D, regulation, and access. And today I will focus mainly on the first 3 pillars. So for pipeline products, we develop TPP, and we are also exploring regulatory efficiency to arrive at the generation of an optimal data package to support regulatory review. For antibiotics that are already approved, we are in contact with the manufacturers. WHO published an expression of interest for PQ. So first of all, we started in setting global priorities for R&D. So we have prioritized antibiotics that have already a pediatric indication, but lack a pediatric formulation. And these antibiotics forms our priority list. We also prioritized antibiotics that are in the pipeline already approved, for which further investigation is needed in children. So the watch list and the priority list are our R&D priorities to address serious bacterial infection in children. And with the project number 2 and 3 that I'm going to illustrate, we addressed the priority and the watch list. So after having established global priority, we focused on the priority list. So antibiotics that were missing an age-appropriate formulation that should be developed ideally in the next 3 to 5 years. So for these antibiotics, we developed target product profiles that are already available on our website and can be downloaded from the QRT code that is on screen. So we developed TPP for azithromycin, nitrofurantoin. We didn't develop it for amoxiclav because according to the experts supporting our work, the amoxicillin clavulanic acid ratio is not influenced by the doesn't influence the likelihood of adverse events. So no further dose formulation were deemed required until we will have a new PKP data. We also focused on the watch list to accelerate optimal approval of antibiotics for pediatric use. Our watch list contains 2 products, Ceriprime-Tazobactam, as Peter said, is on a clinical hold at the moment with US FDA, and we also have Sulbactam-Durlobactam that was approved with US FDA in 2023. WHO hosted a series of regulatory consultations with 2 purposes: to foster convergence in regulation and to guide the generation of an evidence-based data package to support the regulatory review from regulators, but also for policy and clinical practice. So to foster convergence in regulation, we reviewed the current regulatory requirements, focusing in the initial phase of this project on 2 main geographies, North America and Europe. And the ICH E11A guideline is the framework that is supporting pediatric extrapolation. However, we noticed that there are differences in the approach to learning, and this often results in 2 distinct pediatric development plans with further cost and burden for the small and medium enterprise that are still active in antibiotic development. We were also keen to hear during this consultation the challenges from drug developers, and these challenges are really multi-phased. The biggest challenge for sure is the lack of funding. So when companies arrive with an adult indication, as Ramanan said, the stock price drops, but also they've exhausted all the financial means to start a pediatric investigation. But there are also some specific factors that hamper development in children. There are some scientific challenges, for instance, the distinct disease manifestation across the pediatric age groups, or the different organ maturation that has an impact on metabolism and exposure. There are some ethical issues, for instance, the parental hesitancy to enroll younger children in clinical trials. There are also some other regulatory issues, such as the difference in the approach towards the needs for single dose versus multiple dose studies. But after having explored and unpacked the challenges, we try to focus on the solution and recommendation to guide the generation of this robust evidence-based data package for regulatory review. And we were really keen to hear the perspective of clinical researchers. So if we have, under the assumption that the same exposure in adults and children results in the same safety profile, a simplified pre-marketing safety data package can be used. This should, of course, integrate and enhance post-marketing pharmacovigilance data with data from registries or real-world evidence to monitor adverse events that emerge with the routine clinical use. So this paradigm shift involves several steps. For sure, the validation of the tools to be used by regulators, the quantification of uncertainties and the gaps in knowledge, and the synthesis of the available evidence. During this work, we proposed a framework for global alignment. So when we have a similar disease course between adults and children, extrapolation of safety is generally acceptable and efficacy as well for well-established class. For first-in-class, the extrapolation of safety and efficacy is allowed in some geography, by Guinea in particular, but while the US FDA usually adopts a more risk-based approach, considering also the unmet medical need. When the disease course is different between adults and children, extrapolation is usually not possible, and so a randomized clinical trial is generally needed. So after these pediatric regulatory work streams, our efforts will continue in the pediatric space at WHO with the development probably starting from next year by guidance for the generation of evidence to inform regulatory submission, clinical practice, and policy. And please stay in contact with me because we need pediatricians with an expertise in infectious disease. I'm also presenting these slides, this final slide, on the behalf of the GAPF accelerators with my colleague Martina Pernazzato, that is leading this effort. They presented the 2025-2030 strategies last week during our 78th World Health Assembly to contribute to global access to medicine for children and support universal health coverage. And the GAPF accelerators is supported by a number of partners, and many of them are there with you today in the room, and many stakeholders. So we really are looking forward to continue this effort between the MR division and the GAPF accelerator. Thank you so much for listening to me today.
Thank you, Valeria, and thanks too to the audience. That wasn't that easy to hear, so great. Could the speakers all come up, and we can open this up for discussion. So we've heard a lot. You guys can come and sit here if you want. We do have a description of the problem. As you all know, a vast number of young babies that previously would have died are now born in hospitals instead of outside of hospitals, but hospitals that are under-equipped in terms of infection control, so they run the gamut of enormous morbidity and mortality, and it's caused mostly by resistant gram-negative infections. And you've heard about some of the approaches to this, some of the exciting work finally happening in this very large trial. And so, Peter, I'm afraid the first question is for you online, which is asking why it takes so long and why it costs so much. So perhaps you can start to address those issues for Samuel Channon Wells asked the question.
I think there can be a very long answer, but I will try a short one. Why does it take so long? As I said, we did the observational study where we had to identify the sites, enroll more than 3,000 babies in the study, collect the results, analyze the results, identify actually the treatments that we think are appropriate as new treatment regimens. We did hollow fiber studies with fosfomycin and flomoxef to see whether they actually work synergistically with amikacin. And then we started the new SEP1 study with a new clinical trial design, which we call practical, personalized, randomized controlled trial. There's a publication by Sarah Walker in 2021. So this is also, it's not a standard run-of-the-mill trial, but it's a new trial design. So you need to also convince ethical committees that they actually approve this new trial design. So there were many steps that actually we had to do, and now, as you saw from the presentation, we are opening many sites in different countries, and every different country, of course, has their own requirements also that you have to fulfill. Why does it cost so much? I actually think this is pretty cheap. Huh? I mean, if you think 50 million for 2 studies with more than 3,000 babies, I don't think that anybody can do this cheaper.
So, I mean, the one thing that I was encouraged to see is that cefiderocol is going to be part of that design, and I think that we would definitely like to see, because for all the use of the other agents, none of them are currently being widely used in rich countries, and I think it's critical that we'll deal with the costs of these drugs down the line, but I think it's critical that these babies get access to the standard of care therapies as well. Right, we've got opportunity for anybody in the audience to come and ask questions. Ron always has a good question, and here we have a question there. Do you want to start?
Yeah.
Okay,
Yeah,
off you
please.
Hello, my name is Asha Bowen. I'm from Australia and the president of Whispered. I was quite intrigued by the presentations and really enjoyed thinking about how would we strategically combine all of your talks on a daily basis on a ward round, and wondering about that kind of integration between antimicrobial stewardship and the issues that were raised with that between access and conservation, the issues with vaccination, and then I guess the infection prevention and control and the WASH approach. And I just wondered, for many of us as clinicians in the audience, if any of you had any ideas about the integration and clinical care of those 3 approaches.
Padmini?
Yeah, thanks for that question. I mean, I don't think that there's any one pro forma answer. I think the vaccine part is perhaps the most straightforward. Vaccines, you know, at a level different from a direct clinical care, there needs to be advocacy from clinicians to make sure that the national programs are approving and recommending vaccines, and that we target vaccine hesitancy where and when it exists, so that a fully vaccinated child will, of course, have decreased risk for some of these key infectious disease pathogens. In terms of treatment, I mean, so actually I'll take a step back. For infection prevention and control, it's a really difficult issue because it doesn't rely only on clinicians. It's really a whole of hospital systems issue. Having that advocacy within the infectious disease community, making sure that there are nurses who are trained for that, I think can make a difference, and having metrics to actually measure what is being done can actually make a difference. And In the United States, the thing that actually made a difference in healthcare-associated infections was changing the way hospitals were reimbursed based on their rates of healthcare-associated infections. Now, whether that applies to all countries or not, it's hard to say. And then on treatment, and I invite others to comment as well, I think it's a challenge, a different challenge in different settings. In some settings, it may be a challenge of, do you have the antibiotics available, or is there only one on the formulary? And that makes the decision really straightforward, or takes the decision away from the clinician who would, I imagine, rather have that decision-making power. In other places, to be able to have clear guidelines, and we need better data. The neonatal sepsis treatment guidelines that were updated from WHO in 2023 still recommend ampicillin and gentamicin for hospital-onset neonatal sepsis. And that's really what drove us to support the NeoSep trial, is we actually need better data that is not just an A versus B trial to provide the recommendations for the types of sepsis we're seeing nowadays.
Maybe I'll just ask Ramanan a question, because one thing that is extraordinarily striking to me is there are vast parts of the poorest parts of the planet where you will find carbapenem-resistant strains circulating in the community. And the reason is that these drugs are not used in the community at all. They're used in the hospital only, and the feces of the patients in the hospital mix with the drinking water of the community. And therefore, these organisms spread enormously in the community. So sanitation becomes a key part of infection control. Do you want to address that? And then we'll go to your question over there in a minute.
That's a great point, Keith. So on the one hand, as Padmini said, the standard treatment guidelines are for AMP and GEN for treatment for sepsis, but that's not what's actually happening in most places. In a study that we're sort of wrapping up in Tamil Nadu in India, it's a sort of, it's probably the per capita GDP of Thailand, for instance. Something like 80% of these children get on carbapenems in the first, couple of weeks of life. And it's without any access to diagnostics. So I think, implicit in the question is, it was asked by someone from Australia, which has, access to a very different set of infection prevention control, barrier protection, and also diagnostics than you would see in the vast majority of places where these drugs are being used. And I think we're in a critical juncture where the way in which we think about appropriate access to antibiotics goes through a particular kind of definition of stewardship, which is mediated by diagnostics and in a particular pathway. And you have to remember why we did this in the West. It was both because those tools were available. It was also a cost containment issue. That's where it first came from, right? You didn't want clinicians to be prescribing, very expensive antibiotics. So you say, okay, you put these things on formulary, that's what happens. I think we need to really rethink stewardship in LMIC settings to be focused on access, but not having to rely on this mediation through diagnostics, which I'm not pessimistic about in the sense of thinking it's not important, but pessimistic about in the fact that we have study after study showing that even when they're available, they're not actually followed. There's not necessarily the clinician capacity to follow through on them. And so I don't think we can take this model of how we're using antibiotics in high-income countries and drop that into LMICs and say we just need to train people. That is not going to happen. I personally think that there's tremendous scope for AI and other mechanisms, and I suspect that in 5 years from now we'll be having a very different conversation about these sorts of things. But the current approach is just not sustainable in any way. It just has to be very different.
Thanks. Yeah, your question.
Hello?
Yeah.
Tobias Senbaum from Berlin, Germany. Thanks for all the 4 interesting presentations. You all illustrated that the burden in children is sort of unrecognized and many have not paid enough attention to the topic. And as we realize within many other pediatric topics in health and in other issues, how what is your recipe to address this topic to politicians, to stakeholders and others who are important in the field to support progress to overcome antimicrobial resistance and access to the drugs? What would be your recipe to move forward? So the question is to all of you.
So, may I just give a very short, short answer? I'm not sure if there's one recipe that we can use for all these questions. We have so many issues in the world that's really hard to get through to politicians. And I think that's just the main question that we have and other sectors in medicine also. But we have to keep trying and just keep doing what we are doing. But I'm not sure about the recipe.
Yeah. Peter?
Yeah, I think a very interesting question. I remember I pitched once a project to the Sherpa of Germany when they were the G7 presidency and on AMR, and we said, oh, there will be 50 million people dying by 2030, or I can't remember. And then they said, oh, you know, just before you there were the climate change people, and after you they're the ocean preserving people, and they're all going to tell us that we're all going to die if we do nothing. So I think these numbers and competing with the numbers is in my experience with politicians and parliamentarians not often very persuasive, because everybody is doing it. But where I can see where we can get buy-in is if you actually come with solutions and say, yes, we have a problem and the problem is bad, but we also know actually what we want to do. And I think what Ramanan just said, I think we know that there are, and also what Padmini presented, you know, there are vaccines, we can actually develop more vaccines, we can invest in IPC, we can invest in WASH, we know absolutely well that this works. We can actually do things like the NeoSEP study, so it's not that we don't know, you know, what we would do. And I also believe that in comparison to other problems, the required investment is also not outrageous. So my experience is if you talk to politicians, if you propose them solutions that are affordable, that is the best strategy.
So we have a number of questions online. I'll address two of them. One is pointing out that much of the problem in neonates is related to prematurity. And obviously if we could reduce prematurity, and there's certainly a huge excess of prematurity in poor countries, that would go a long way. Of course, that is an active area of interest at the foundation where we are trying to find solutions, but it's not an area where I'm very optimistic at this stage. So it leads into the next question, or many lead to these questions. I guess it's inevitable we get a question about AI. And there's no doubt there's enormous potential, especially in low middle income countries where there's a huge deficit of clinical care, for AI to produce algorithms which will help doctors and nurses to prescribe better and to treat better. And so we are also very, very excited about the possibilities of AI to bring better protocols and make them more available in these poor parts of the world. Now there's a specific question for Padmini. Vasiliki Papavengelu is a little upset that you did not talk about influenza vaccine to reduce AMR, so could you do that?
Yeah. We only had 13 minutes, so I could only pick and choose a few vaccines, but I would agree with the person online that there are other vaccines beyond those that that I had a chance to list that do have an impact on both reducing disease as well as antibiotic use. Influenza, like RSV, is a very common viral respiratory illness, and I think, you know, in terms of actually potentially making impact on antibiotic use, these vaccines that target very common viral illnesses potentially have a greater impact than some of the other vaccines that would target bacterial pathogens that are ultimately have a lower incidence. For influenza, there are great data, mostly from high-income countries, that the use of influenza vaccine does result in significant decreases in antibiotic use. That reduction in antibiotic use varies with how efficacious the vaccine or effective the vaccine is in a given year, as well as the level of coverage, which is not surprising. And then there have been maternal influenza vaccine trials that have shown that if you give a pregnant mother the influenza vaccine, you do see a reduction in influenza as well as all-cause hospitalization in the young infant in the first 6 months of life. And I would imagine you'll also therefore see some reduction in antibiotic use as well.
So we have more questions. I'm going to come to you in a minute, but just the two more online. One expressed surprise, and in fact, when you see the data, one is surprised at the amount of antibiotics that were used for diarrhea in your study, Ramanan, and I guess Padmini can address it as well. That seems to me an area where AI could go a long way to improving treatment options. But then somebody noticed, Padmini, that there's enormous gender difference in mortality in the extremes of age for Klebsiella, and it's mainly males. So she wanted to know if we know anything more about that.
Yeah, I mean, I do think that we see some differences in prematurity, and those differences in prematurity are by gender are also then put these babies are at greater risk. In fact, in Klebsiella, it's almost entirely in premature infants that we're seeing the Klebsiella neonatal sepsis, and therefore the burden of Klebsiella is really a reflection of the burden of prematurity. So it comes back to Keith's earlier point, or the point that someone online commented, that really getting it at the root of the problem in terms of trying to reduce and prevent prematurity is perhaps the most important solution we can go for, that is not targeted at a specific pathogen.
We have a question down there.
Yeah, Michiel van der Vlier from Utrecht, the Netherlands. I've also a question for Dr. Srikantiah. I myself am working also on antibodies against neonatal sepsis, and I was wondering, referring to the Klebsiella and prematurity and to the 10 pathogens that are relevant, which would make a lot of maternal vaccines necessary, do you see in a bit further future a place for a cocktail of antibodies against these pathogens?
Yeah,
you want to address that fine?
Yeah, it's a great question, and I agree with you actually that something that can be used, a maternal vaccine means that we are targeting an entire cohort of a birth cohort to prevent Klebsiella in a fairly small proportion of those of that birth cohort. Whereas if we can target premature infants at birth, it could be a more efficient approach. The challenge with monoclonals right now, and I'll just give you the example of Klebsiella, is that we're struggling in a vaccine strategy to be able to come to a reasonable valency at a 5 to 10 valent vaccine to be able to cover most of the serotypes that cause infection. A monoclonal antibody, I think, would be difficult to achieve that level of protection. And then if you come to a cocktail, the costs currently with monoclonals would truly be cost prohibitive, particularly in low and middle income countries. I think as we look to the future, there are some strategies that our partners in the discovery program at the Gates Foundation are working on in terms of things like multibodies that allow a combination of different targets. Our RNA enabled monoclonal antibodies may also help to reduce that cost. So having something that could target either multiple serotypes or even multiple pathogens, and identifying what those targets are. So right now we're focusing on polysaccharide antigens. Perhaps there are others that are a little bit more broadly based.
So Valeria, if you had a wish for a single change in regulatory authorities that would help to get more access for new antibiotics in young children, what would that be?
Thank you for this question. Actually, it is the scope of our current project to try to foster convergence in regulatory requirement. I think this is a really relatively small efforts from major geography at the moment, but I'm sure that other geography will be willing to jump on board. As I mentioned in my presentation, the numbers of pediatric studies are already duplicated at the moment in some instances. And if you have to develop some products also for neonates, the number of studies required will be increasing. So my wish and the looking forward would be to have really a unified pediatric development plan, kind of plan harmonized across different geographies that developers can follow in their development plan for new antibiotics.
Thanks.
And this will surely reduce cost and probably increase also global access.
Thank you. Here's a question.
Shai Ashkenazi, a very nice session. Just my idea from my experience, we have to try to think outside the box. In other words, there are many new innovation approaches of non-antimicrobial therapy of infectious diseases. So this is the way, I guess, because we are probably reaching the end of the antibiotic era with the resistance outpacing the development of new antibiotics. Now we know a lot about the infectious processes. We have now agents that can prevent the adherence of bacteria to foreign bodies and to the mucosal surfaces. We are able to do it. We have anti-quorum sensing to prevent infection on foreign bodies, antitoxin production, so antiseptics for bacterial infections, like the alcohol lock for the intravenous infections. So I think we have to think more and more outside the box with the title of non-antibiotic treatment of infectious diseases.
I think that's right, and I'm going to let Ursula talk to that some more. I certainly know there are very exciting ideas around interrupting biofilms, for instance, and if they could be more general. But the way I want to also move this is to say that I had a discussion with Ursula earlier, and of course, if we prevent one bug, it's not going to really solve this problem. We need solutions which have a broad preventative application, and I think that's what you are talking about.
It's very broad. I mean, the biofilm, we know now there is anti-quorum sensing that relates most biofilm formation. Yeah. We know that.
Yeah, that's right.
So also a very short answer because it's almost the end now. We are beyond our time. I think there's still room for new antibiotics, not with just modification of old classes, but new, completely new targets and new ways of mode of action. But we have not enough power to explore all these ways. We are not putting enough effort into these different ways. And of course, we need to think out of the box, and that has been done already since 30 years. As with every innovation, it's very difficult to prove that they work. And if they are too small in target only very few pathogens or very specific patients, then it's extremely difficult to show this effect. So it really depends on the kind of innovation. I think they need to target a really wide patient populations and need to be inexpensive and also be usable without great infrastructure, healthcare infrastructure. So these are the hurdles, but still we need to have a lot more research in this area.
We do have time for one last question.
Thank you, Phil Britton from Australia. And I acknowledge that I'm from a high-income country, so that might be biasing my question. But if I ask a question about the conserve pillar, I want to challenge the comment that diagnostics might not be critical to that. I can't see how we will conserve new drugs or new combinations without better diagnostics to know which child really needs them into the future. So my question is, one, do you agree with that proposition? Number two, what kind of diagnostics might be required around the world in order for them to be delivered at scale? I mean, I'm thinking Gene Expert, for example, for tuberculosis, which has stopped clinical diagnosis of resistance and use of ineffective regimes for extended periods of time. But is that the kind of diagnostic that we might need?
Yeah, I think nobody can really argue against that argument. The problem, of course, is cost, and at the moment, you know, in poor countries, all that's affordable are kind of lateral flow techniques, and so it's very limited what's available. But surely you're right. In the long term, the answer to more appropriate antibiotic use has got to be to learn to direct it better. I think that we're done. I want to thank the audience. I want to thank my co-chair and all of the speakers. Thank you guys so much. I'm glad. Thank you, Kenneth, for getting the noise down reasonably. And I think we've had a very broad-ranging discussion on the issues of AMR, particularly focused on young kids. And hopefully there'll be better news in the future. So thank you.